So let's move on to describe gene cloning and the polymerase chain reaction. So first we'll do gene cloning. So the key word to focus on with gene cloning is isolating and making copies of a gene. For gene cloning, we'll talk through the process and then we'll talk about why you'd even want to do something like this. So to clone genes, first you'd isolate the gene of interest. So let's say in a human cell that you're interested in a gene that contributes to, let's say some healing process in your body. So you isolate this gene from a human cell and you cut that gene out of your cell of interest and then you paste it into a piece of bacterial DNA called a plasmid. And this is a special piece of DNA that only occurs in bacteria, and they're really useful in this instance of biotechnology because they can allow us to make copies of a gene. So you paste that new piece, that gene, into the bacterial plasmid, and then you're able to put that plasmid into a bacterial cell. And we know bacteria can replicate exponentially faster than mammalian cells. So you can get a large amount of this gene of interest really quickly. So that's what that's called right here. Recombinant DNA is a piece of bacterial DNA that's had a new gene pasted into it for the purpose of cloning or making copies. So the steps of gene cloning are isolation, ligation, transformation, and selection. So isolation is the first one, where you cut out the gene from the original genome that you were interested in. So you use an enzyme to cut or break the DNA at a very specific base pair sequence and isolate that out from your organism or cell of interest. And then ligation, the next step, you take that gene and you combine it with the plasmid from the bacteria and add some more different enzymes that are able to paste it back together. So this circle's broken, and then ligation is the circles put back together. And the result is this recombinant DNA. So this recombinant DNA is a combo of your gene of interest plus the bacterial plasmid DNA. And the next step, transformation, is where you put this recombinant DNA and insert it into a living cell, usually a bacterial cell. And this is an example of genetic engineering. It's inserting a new DNA into a different organism. So different DNA from another organism into a new organism. And then the next step, selection, you grow that bacteria up, and there's different ways you can test for the DNA that you want to be present there. So you want to grow only the cells that contain your gene of interest. So that step's called selection. And then you're able to have those copies of DNA that you selected for and use this for purposes in biotechnological research. So for example, gene therapy is a common reason that you would use gene cloning. So say someone has a mutation in a gene and they're not able to metabolize certain nutrients in their body. And so that can result in disease. And there's different forms of gene therapy where you can clone a correct non-mutated gene that person's missing, and then give it to that person as therapeutic treatment, and they're able to be more healthy. So gene therapy is an application of gene cloning. And then next, let's talk about polymerase chain reaction, or PCR. So the key word to remember with PCR is making many copies of a gene or DNA segment. And the way you do that is through changing the temperature, so heating and cooling, and plus the correct ingredients you need to make DNA. So PCR is making many copies of a gene, which is called amplification. Well, first off, let's take a step back. Why would you want to make many copies of a gene? So you need a large quantity of DNA for scientific instruments to be able to detect it, and so it's useful in a lab setting. So some examples of why you would want to use PCR to amplify a gene is sequencing. So sequencing is where you learn the exact sequence of letters or bases in a stretch of DNA. And that can be important for different applications in research, like figuring out what genes correspond to which traits. This is also used in ancestral DNA applications. Another example of why you'd use PCR is to detect the presence of a gene when you're trying to detect an infection. So in the early stages of an infection, you can take samples from a patient and amplify their DNA and see if bacterial DNA is present and see what sort of infection they might have. Another example that you've probably heard of is like forensic profiling. So from a crime scene, they collect small samples of DNA and then they're able to amplify it up and figure out who that DNA belongs to using PCR. So now that we've talked about why you'd use PCR, let's talk about the steps. So PCR involves 3 main steps. So denaturing, annealing, and extending. So during the denaturing phase, denaturing means unwinding or unraveling. So we know DNA has 2 strands wound up in a double helix. And when you increase the temperature, when you heat things up, DNA unwinds. So the first step is you heat it up and denature the DNA. Next step is annealing. So this is reversing that step. You bind the DNA back together, but in this step, you've added in an extra ingredient called primers. So the DNA doesn't just bind back to itself together, it's now bound to these little sections of DNA called primers. And these are designed to bind to the DNA in a specific spot, so you know which section of the DNA you want to make copies of and you want to amplify. And the primers are like the starting line. And then the last step is extension. And this is where you add an enzyme. It's called DNA polymerase. And this acts on the DNA, starting at the primer, to synthesize new DNA. So make a new copy of the original strand of DNA. And then you repeat this over and over and over until you have many copies of the original gene. You need the right ingredients to carry out PCR. So you need the DNA template, the original DNA strand to be copied. You need those primers, those short stretches of DNA that initiate the PCR reaction, and they're designed to bind to the DNA right at the section that you want to copy. And then you need the DNA nucleotide bases. So these are the letters that are the building blocks of DNA. So A, C, G, and T, if you remember from the previous objective. And then you need the enzyme, the DNA polymerase, that will actually do the construction work of building the new DNA. And you need a buffer. It's the liquid or the mixture that all the ingredients sit in to ensure the right conditions for the reaction.